So this whole thing on your Canvas webpage, the way that things are going to be working from now on is, and again, it's going to be a work in progress because I'm still figuring out exactly how it goes, but the modules are going to be the way that we're going to be able to kind of see where are we, what assignments you've got going on, what things are due. Like for instance, one of the things I'll talk about when we get to lab time is we're going to, since we can't turn in our lab books, we're going to actually be scanning parts of our lab and submitting them. And so there's going to be things in this module here that guide you on how to submit things. There's going to be little, again, as this keeps going, there's going to be links to things that'll help you with your studying, blah, blah, blah. But again, it's a work in progress because I'm figuring this out as we go as well. At any point that you're having like, oh shoot, we're getting this feedback again. At any point you have questions about anything, please just email me and I'll do my best to make sure that we get things straightened out. What else? In terms of testing, we're not sure exactly how that's going to work yet. So we're really hoping, kind of keeping our fingers crossed, that there is going to be like in-class meetings again, like after April, and that we'll finally we'll be able to do actually real tests and all that. So at least for the next couple of weeks, while we're still trying to see how things are going to look, we'll do online quizzing and stuff like that, but we're not going to have any like major exams yet because we're still not really sure what that would actually look like for a class like this. Does anybody have any questions?
Due dates, are they posted on Canvas?
They'll be posted on Canvas, yeah.
Okay.
And what else?
Scanning of, do we take pictures of the notebook or do we have to download a scanning app?
It's a scanning app that you'll download on your iPhone, and I'll talk about that when we get to the lab part. So so what we'll do now is we'll see what it feels like to actually do kind of lecture stuff, and I will, and again, as we do this, after we're done with this, because one of the things they've also talked about is people, at least, people might have like kids that are not at school, or, depending on what your schedule's like, if you don't actually, hopefully the classes will still be going pretty regularly, but if you don't end up in a class, each time we're done with this, I'll kind of clean it up and I'll upload it. So you'll have access to all of this stuff online as well. All right, so let's try this. I'm gonna go and share my whiteboard here. All right, so last we met, we had talked about, we were finally starting the peripheral nervous system. And let's get a smaller. So if you remember, there was like the somatic versus the what? What was the other one?
Autonomic.
The autonomic. So the somatic was the one that we were talking about right at the very end of last class when we were last all together. So there we looked at how the somatic motor was connected. We had like the brain, the spinal cord, and the target, if you are looking at the somatic motor system, is always going to be some skeletal muscle because it's voluntary. And we talked about if you're connecting from the brain and spinal cord going out to a skeletal muscle, it's always going to be just one single neuron who has his cell body in the CNS, and then an axon that goes all the way to the target muscle and has its little neuromuscular junction there. So there's, for the somatic, there's only just one neuron with the cell body in the CNS. And do people remember what kind of neurotransmitter is released by the somatic motor neuron at the muscle?
Acetylcholine.
Yes, acetylcholine, exactly. So acetylcholine is the neurotransmitter we release here. And then we also talked about the special kind of receptor that you have on the skeletal muscle. And it was the one that's always going to be excitatory, which are the nicotinic cholinergic receptors. So that is, hold on. I'm actually realizing I can probably do some, like, let me try this really quick. If I go this and go here, I could say, back. I can spell it. Yeah, so that probably looks a little less wonky than my writing. So that is the somatic. So that was the one that we were covering. So what we're going to do now is we're going to go and look at the autonomic, which is going to be a little more complicated. So let's I need to move off of this screen. So now we're going to go to the autonomic nervous system. And so again, the targets, what are the targets going to be when we're looking at the autonomic? What kind of things get activated by autonomic motor neurons?
Inner organs.
These are going to be like organs, like your heart. It's going to be, what other kind of things? Glands, going to be turning on your glands. This is also going to be like smooth muscle. You know, the muscle that you don't control voluntarily, like the diameter of blood vessels and the, you know, diameter of the pupil and things like that. So there's a few things that are very different about autonomic motor, the way it's connected. If here's the brain and spinal cord again. Brain, oh my God. Pretend this is the brain, spinal cord, and let's say here is some target, like your heart. So one thing is there's always going to be two neurons. There's going to be one neuron that starts in the nervous system and comes out, but it doesn't go all the way to the target. So this would be like my target. And then there's going to be a second neuron that then continues and goes all the way to the target. So this right here, whenever you have cell bodies of a neuron that's not in the CNS, that's in the PNS, we call that a ganglion. So ganglion is the cell bodies outside of the, or in the PNS, I should say. So in the autonomic motor, there's always going to be ganglia. You know, where the ganglia are going to be different depending if you're looking at sympathetic or parasympathetic. And so we'll talk about that in more detail. But it's going to be important that there's going to be two different neurons. There's going to be a first neuron. We usually call this the preganglionic neuron. And then the second one is obviously going to be the postganglionic. So that is how they are similar. And remember, autonomic motor is going to be sympathetic as well as parasympathetic. So both sympathetic and parasympathetic are going to be similar in that, again, they're targeting organs and glands and smooth muscle. They use two neurons, the first neuron, preganglionic neuron, that leaves the CNS, but then a second neuron that arises from the ganglion and continues on to the target. I'm looking at my drawing and I feel like this looks like a kindergarten drawing, but it's good enough. So now what we're going to do is we're going to look at how sympathetic and parasympathetic are different from each other. And they're different from each other in some pretty major ways that are really useful clinically, because you can actually target one or the other using different kinds of drugs. So if you're trying to speed up the heart or slow down the heart or keep the heart from speeding up or keep the heart from slowing down or anything, you can actually take advantage of the fact that sympathetic and parasympathetic have some pretty profound differences in how they're wired up. So let's do that. Let's, here, I'll say, sympathetic versus no, I'm actually trying to keep the color coding going here. So now I'm going to try. Okay. Alrighty, so sympathetic versus parasympathetic. All right, so obviously one big difference we've already kind of talked about when we introduced them was sympathetic tends to be the more excitatory. It's the one that speeds up the heart. It's the one that dilates the pupils and gets you ready for danger. Parasympathetic was the one that slows down the heart, you know, increases digestive processes, will make you salivate more, things like that. Another big thing is if we look at, you know, the brain and the spinal cord, if we look at sympathetic, the preganglionic neurons arise from the thoracic and lumbar regions of the brain and spinal cord. So we call it the thoracic. So hold on, somebody's got like a feedback loop going here. So let me go into my manage participants window and see if I can fix this. Okay, let's see. So thoracic and lumbar. So thoracic would be like around here. Lumbar is lower back. So sometimes we call this a thoracolumbar outflow. Or I want it to be red. You're doing thing. Thoracolumbar outflow. Whereas if on the this is going slower. From the parasympathetic, it's starting from cranial nerves and from nerves coming out of the sacral region of the spinal cord. So this is going to be what we call the craniosacral outflow. So that's one big difference. And the like maybe 70% of the parasympathetic is actually coming from one particular cranial nerve called the vagus nerve. It's called cranial nerve number 10, which is the vagus nerve. But not. So, race. So the vagus nerve it's the only one of the cranial nerves, the nerve that comes out of actually originates at the brain, and it leaves the head and goes all over the body. It's the one that goes to your heart to slow down your heart. So that's one thing. Thoracolumbar outflow versus craniosacral outflow. Another big difference is the length of the different neurons. So when we're looking at sympathetic, there's going to be a short first neuron, and a long second neuron. Whereas when we go look at the parasympathetic, it's going to be a long first neuron and a short second neuron. And so I can kind of illustrate that. Like for instance, if you were, let's say you're going to the heart and you're on the sympathetic side, there's like a short first neuron, a ganglion, and then a long second neuron that continues all the way to the target organ. If you're on the parasympathetic side, let's say here's the heart again, you're going to have your first neuron goes a really long way. The ganglion is right near the organ, and there's just a very short second neuron. In fact, the ganglia in the parasympathetics can sometimes even be within the wall of the target organ. They call them like terminal ganglia because terminal kind of end of the line. They're also sometimes intramural ganglia, means within the wall of the target organ. Whereas when we're looking at the sympathetic, the ganglia are usually right along near the spinal cord. So, you know, we're going to talk, you know, the sympathetic chain ganglia, there's these ganglia up and down all along right next to the spinal cord, and then long second neurons that go all the way to the target organ. This might be a good time for me to try. Let me see if I can do this. Booby-doo. Here, I'm gonna share All right, so here, this is like a picture from your text that kind of gives you a sense where you can see, in fact, I can use my point, I can use a highlighter, and you can see like here's sympathetic. Sympathetic, we've got, this is the ganglia, which are right near the spinal cord, and these are the first neuron. Then the second neuron continues all the way to the target organs. And also notice that the outflow is coming from the thoracic and lumbar region, whereas for parasympathetic here, we have the outflow, the initial neurons coming from the cranial nerves, which are coming from the brain, and the spinal nerves coming from the sacrum, and then really long initial neurons that go almost all the way to the target, and then a second little neuron, which you can even barely see on this slide. So this is kind of giving you a sense of one of the differences here and just how they're wired. But then the other big difference is what kind of neurotransmitters are being released at the target. So at the ganglion, this first synapse, which is happening here, is just kind of like a relay. That one is actually just going to be acetylcholine released to nicotinic cholinergic receptors, kind of like what we saw at the skeletal muscle. So this first synapse at the ganglion is just like a relay. It's this excitatory thing that's just going to keep the message going. But the interesting difference that we're going to see is what happens when the postganglionic neuron, the second neuron, reaches the target organ. So that's what we'll look at now. So let me go back to this. Share whiteboard. So again, we're going to be looking at sympathetic versus parasympathetic. So sympathetic usually, I'll say usually because we'll see there's some exceptions, releases either norepinephrine, which we usually just abbreviate as NE, or epinephrine, which we usually say is Epi. So this is like the second neuron. The second neuron usually releases norepinephrine or epinephrine. And those, if you're in England, you'd call them noradrenaline and adrenaline. Epinephrine is just another word for adrenaline. And parasympathetic, on the other hand, the second neuron always releases acetylcholine. And the difference between parasympathetic and the somatic, though, is even though they both release acetylcholine, the target is going to have the different kind of receptor. We talked about there's muscarinic and nicotinic cholinergic receptors. So in the parasympathetic nervous system, the target organ has those other kind, the muscarinic cholinergic receptors. Muscarinic cholinergic receptors. And if you remember, does anyone remember how muscarinic cholinergic receptors are different from the nicotinic?
As a second messenger.
Say it again?
Second messenger?
Yeah, exactly. They're second messenger mediated. So these have lots more options for what the actual response is going to be. So if you operate a somatic motor neuron, it's always going to excite the target skeletal muscle. But when you turn on a parasympathetic autonomic motor neuron, you don't really know what the response is going to be at the target. Sometimes it could excite it, sometimes it could actually inhibit it. So there's much more variability in the response when you release acetylcholine in the parasympathetic motor. Sympathetic, let me go to red. The targets have adrenergic receptors. Adrenergic. There's a variety of different kinds. We're not going to go into all the details now. There's like alpha 1, alpha 2, beta 1, beta 2. Like, you know, when people talk about taking beta blockers to help keep their heart from speeding up if they have like heart blood pressure, beta blockers are blocking beta adrenergic receptors. And, you know, so one of the nice things about having this very distinct neurotransmitter release for parasympathetic versus here. Oh my God, hold on. This is Bagheera.
Hi, kitty.
He's like walking all over my thing here. Let me get back. All right. Hold on. All right. Sorry about that. So one of the big differences is this adrenergic means you can, if you want to block sympathetic, for instance, for high blood pressure, you want to keep the heart from speeding up. You want to keep the blood vessels from constricting and increasing resistance. You can give things that are antagonists to adrenergic receptors. So adrenergic antagonists would block sympathetic activity. If you wanted to increase sympathetic, someone's having an asthma attack and sympathetic dilates bronchial passages, you can give them an adrenergic agonist, something that activates adrenergic receptors, and that would help them dilate their bronchial passages. So there's a lot of different drugs that are specifically designed to either activate or inhibit sympathetic response with adrenergic agonists and antagonists. And these different subtypes of receptors are localized on different areas, so you can actually be even more, kind of have more finesse about exactly how you're modulating the activity, not just if you're increasing or decreasing, but like what parts of the body are being affected. Parasympathetic as well, like if you're trying to, like a classic example, if you're at the eye doctor and they want to shine a light in your eye and look around inside your eyeball, typically if you shine a light in your eye, your pupil constricts. And that would be parasympathetic if you make your pupil constrict. So little drops they'd give you would be antagonists to the muscarinic cholinergic receptors. So then when they shine a light in your eye, the response would be send a message to the smooth muscle to constrict. But if you have an antagonist to those receptors, then the muscle does not respond, and so the pupil stays dilated. So there's lots of different drugs that work by activating or inhibiting one or the other. Sympathetic does, when I said it usually releases norepinephrine or epinephrine, there are some exceptions. Sometimes releases acetylcholine. Specifically, like an example is sympathetic is activating your sweat glands. Your sweat glands, and at your sweat glands, it actually releases acetylcholine. Like for instance, so I don't want to make it sound like sympathetic is always epinephrine and norepinephrine, but it usually is. These times it's not are these exceptions. So other things we can add in. So in general, sympathetic and parasympathetic. This is getting sloppy. Are antagonistic, meaning they tend to fight each other. Like sympathetic speeds up the heart, parasympathetic slows down the heart. Sympathetic will inhibit digestive processes. Parasympathetic will promote digestive processes. Sympathetic will dilate your pupils. Parasympathetic will constrict your pupils. There are a few exceptions where that's not happening. So like some exceptions are like sweat glands. Sweat glands are pretty much purely sympathetic. That's one of the reasons why people use, they call galvanic skin response, basically the conductivity of your skin as a measure of your sympathetic response. Like the more sympathetic response you have, the more you tend to sweat, and the more you sweat, the more salt water is on your skin, and salt water conducts electricity. So by looking at the electrical conductivity across your skin, you can actually get a measure of sympathetic activity. It's one of the things they use like in lie detectors. Again, we're not going to probably ever do this in our lab now because we're all online. But we have like those power labs you can actually hook up a patient or a subject to and put electrodes on their fingers, and you kick them, surprise them, and all of a sudden you'll just see the skin conductivity just start going up. Or you ask them a really embarrassing question like, oh, did you have sex last night or something? And they go, bah. And all of a sudden you can just see the electrical conductance of their skin just start going up. Because if you activate their sympathetic, that fight or flight, they'll sweat more and you'll actually see the electrical conductivity go up. Another exception here is the blood vessels are sympathetic. Again, so we're going to talk more about the vasomotor system, like the neurons that operate the smooth muscle in the blood vessels and change their diameter. But those are sympathetic. There are other things that adjust the diameter of blood vessels. There are these like nitric oxide. There are things that will make the blood vessels dilate that are not actually motor neurons. But in terms of motor neurons affecting blood vessels, it's sympathetic. The other place where the example people use of sympathetic and parasympathetic not fighting each other is in sexual response. For normal sexual response, parasympathetic is what actually is responsible for a guy getting an erection. Whereas like the actual ejaculation and all that is going to be sympathetic. So in this case, rather than fighting each other, they're working. One kind of sets the stage, the other one kind of finishes it off. Again, part of the reason why guys can have trouble like having an erection if they're nervous is because they actually have to have this parasympathetic activation for that part of it. So that's usually that's one of the examples that people always bring up of an example of when they're actually not fighting each other, they're working together. So Are there, let me, how do I do this? I'm going to, are there any questions about any of this stuff up to this point?
For the sympathetic nervous system, you said that some release the acetylcholine and this, it would be the sweat glands. What kind of receptor would that be?
That's going to also be muscarinic.
It would also be muscarinic?
Yeah, I'm almost sure.
Okay.
I should double check, but I'm almost sure. And I should also get feedback from you guys. Like, is this, like, I can say, like, for me, it's incredibly, it feels very different. It's kind of tiring. But is this actually, does this work? Or any kind of feedback that you can give me as we go along, or things you've experienced in other online classes, or even like watching like Khan Academies or something. Like, I would love feedback so this is as useful as possible. Like, I do, you know, I'm committed to making sure that we get through this semester and cover all the material you need to know to actually, you know, move on to your next classes and whatever you're doing next. And I would love it to be like as kind of engaging and like as unpainful as possible. So if people can give me feedback on what works, what doesn't work, or suggestions on how to make things feel like more flowy or something, I would totally be into that. And I see Megan is here now. Let me get that here. And Amy is here. And also, oh my God, and Joseph, is Grayley here? Grayley Mazariegos. And Joseph Miller. Joseph Miller. All right, yeah, so in general, we actually have a pretty amazing group here. All right, so if there are no questions on that, I think we're done with the PNS motor. And so now what we're going to do is move into the sensory, which is really fun.
Quick question.
Yeah.
So for the sympathetic, you said the first synapse releases acetylcholine. Is that parasympathetic as well?
Yeah, in both sympathetic and parasympathetic, that first synapse releases acetylcholine. The second neuron has nicotinic receptors, so it's basically like a relay. Like, you don't have, you know, it's basically you send the message to the second neuron, which continues on to the next.
Okay.
Oh, and there's actually one more thing about sympathetic I need to add in before we're done here. So let me do that. Whiteboard. Like Columbo, one more thing. So sympathetic, in addition to releasing, you know, norepinephrine at its target organ through a specific neuron, can also do a more general broadcast, can also cause the adrenal medulla, which is a gland, to release adrenaline or epinephrine, which then goes into your bloodstream and goes everywhere. So can also act. Adrenal medulla. So adrenal gland is just a little, like, if this is your kidney, the adrenal gland is sitting like, with most of the books, sits on top of the kidney like a little hat. And there's two different parts to the adrenal gland. Later on in the class, we're going to be talking about the adrenal cortex, this outer part, which I'm doing in blue, which is releasing different steroid hormones like cortisol, the stress hormone that you're probably having a lot of right now, aldosterone, which we'll talk about, and changing salt reabsorption in the kidney. But the adrenal medulla is the inner part. And it's actually derived from neural tissue, and it releases adrenaline or epinephrine. And this is now a hormone rather than a neurotransmitter. So distributed by the bloodstream. So the epinephrine basically goes into your bloodstream and then goes everywhere. And anything in any cell in your body that has one of these adrenergic receptors that recognizes adrenaline or epinephrine is going to get activated. So if you trigger a release of epinephrine by the adrenal medulla, your eyes will dilate, your heart will speed up, your bronchial passages will dilate. All this stuff will happen all at once. And eventually it will get cleared out, but it takes a little while. So if you get scared all of a sudden, and then you realize, oh, there's no reason to be scared at all, it's going to take a little bit before your heart actually slows down and everything, because you've actually just dumped this stuff into your bloodstream. Epi Pens, probably most people know about Epi Pens. If someone's in anaphylactic shock, which causes all of your blood vessels to dilate, your blood pressure to plummet. If you jam a little needle with epinephrine into their thigh and release epinephrine into their body, it will basically help bring their blood pressure back up, because it's going to talk to all these receptors that are constricting blood vessels, speeding up the heart, things that are going to kind of bring their blood pressure back up in the moment. Epi is just epinephrine, or also another word for epinephrine is adrenaline.
And that could either be, it's both a neurotransmitter and a hormone?
Yeah, typically adrenaline is a hormone, and epinephrine is, and norepinephrine is the neurotransmitter, but a lot of those adrenergic receptors actually bind both. So either one would work. You could either activate the receptor by puffing norepinephrine right at it from a motor neuron, or you could also activate it by dumping epinephrine into the bloodstream. So there are some subtleties to the different beta and alpha adrenergic receptors, but again, for this class, something when you get into pharmacology later, you'll spend more time looking at all of that. So, all right, so that is motor. So now we will move on. Sensory. Also known as efferent. Heck. And remember again the little efferent. These are the signals that arrive at the CNS. These are the messages coming in. And something we talked about in lab, and that's going to be just the ongoing theme for all of our discussion of sensory nervous system, is going to be this idea of sensory transduction. And who remembers what sensory transduction was? Anybody? Was the idea of, I'm going to do this, converting some stimulus. Again, whether that stimulus is light, if it's vision, or kind of deformation of your skin, if it's touch, or maybe it's air vibration, if it's hearing, it's converting some stimulus, into an electrical signal for the nervous system to understand. So this is going to be kind of an ongoing theme for this section here. We're talking about sensory. We're going to talk about, the eye. How does the eye actually convert light energy into an electrical signal? How does the ear convert either like air vibration or, the ears also do your equilibrium balance. How does the ear convert the angle of your head with respect to gravity into an electrical signal? How does your taste bud, or how do your taste buds on your tongue convert the presence of sugar in your saliva into an electrical signal? So that's going to be this kind of ongoing theme. We're going to be looking at all these different senses and seeing which ones do we have and how do you actually convert the stimulus into something that your nervous system is actually going to be able to use. So should also, I can reiterate your the senses are not just going to be the the five senses like your eyes and your ears and your taste and smell. There's a lot of internal receptors that are really important that you're not explicitly aware of. Like your body is constantly keeping track of the stretch of your blood vessels. It's constantly keeping track of, the sugar levels in your blood. So this is both kind of let me do something darker. There's going to be internal receptors. Again, like stretch of blood vessels. pH of your blood. Etc. Just lots of things that you need to keep track of the internal status of your body. And then there's going to be the external receptors, which are the ones that you're more used to, like your eyes and your ears and things like that. So we will be looking at both of these. So let's first just start talking about receptors in general. So receptors. These are the things that actually do the sensory transduction. These accomplish the sensory transduction. And there are a variety of different kinds of receptors. It's good to just explicitly look at them. So, what are kinds of stimulus that we can actually detect? And what kind of things can we actually notice in our outside external surroundings, or obviously that our body needs to monitor inside our body?
Temperature.
Sounds, temperatures.
So temperature. So one kind of receptor are called Thermo Receptors. So we'll talk more about these. These are actually really very simple little receptors. They're going to be part of our general senses when we get to them. What are other things? What other kind of receptors?
Pain.
Pain. So these are called nociceptors. And what is the stimulus? For thermoreceptors, obviously the stimulus is changes in temperature. But what actually is the stimulus for a pain receptor?
isn't like the bending of your skin?
Well, I mean, that could be touch, though. That can actually feel nice.
Is it cell damage?
Yeah, so this would be more like tissue damage or impending tissue damage. And, you know, bending of your skin can indeed lead to pain if it's extreme enough. So in fact, any kind of receptor, if you drive it into the extreme of its range, usually starts getting perceived as pain. But you have specific pain receptors that are turned on by actual tissue damage or impending tissue damage to tell you, like, leave it alone or stop that. So nociceptors, pain receptors. What are other kinds of receptors that we got?
Smell.
The smell. And what is the actual stimulus for smell? What are you detecting? If you smell something, what is the actual thing that you're detecting?
Chemoreceptors.
Exactly. So they're chemoreceptors. So smell, like if you smell like a flower or you smell dog poo or whatever, you're actually just detecting particular molecules floating in the air. The same thing with taste. Taste are chemoreceptors. If you taste sweet, there's actually like simple sugars that are dissolved in your saliva. Chemoreceptors are also inside your body, like detecting glucose levels in your blood or the pH of things, or, you know, so you have chemoreceptors inside and outside. Outside would be smell and taste. Inside, it's just detecting the chemical status of all sorts of things in your body. But wait, there's more. What are other receptors?
Photoreceptors.
There's photoreceptors, totally. The photoreceptors, obviously, your eyes are able to detect light and turn it into an electrical signal that your brain and your nervous system can understand. And there's one other main one.
Meccano Receptors.
Yeah, exactly, the Meccano Receptors. So the Meccano Receptors are going to include things like touch. You know, it's basically some kind of mechanical deformation of things. It's also going to include, actually, it's going to include hearing. Like right now, the reason you hear me talking is your little speaker is vibrating and making pressure waves in the air, which is vibrating your eardrum. And that vibration is being detected by little mechanoreceptors in your ear that you then interpret as sound. Mechanoreceptors are also going to be the stretch receptors in your blood vessels, so you can detect blood pressure. The more stretched out your blood vessels are, usually the higher the blood pressure. Mechanoreceptors are also going to be able to give you feedback into just the position of your body. You know, one of the, you know, I usually think of it as a kind of subtype of mechanoreceptors, but sometimes people just give them their own thing, are proprioceptors. Let me draw that better. Actually, I think they usually say proprioceptors. So these are receptors that give you information about the position of your body. So obviously, it's really important to actually know exactly what your body is doing. Otherwise, you'll just feel like a disembodied consciousness. So you have all of these receptors that are sending information to your brain. These include things like, in fact, we saw one when we did the knee reflex. There's muscle stretch receptors. They're called spindles. There are other ones that give you the angle of your joint. Basically, if you think about, you know, some joint or two bones, if you've been in anatomy, you know, in a synovial joint, there's like the connective tissue joint capsule around that. And then there are going to be little stretch receptors inside the joint capsule. So as you bend the joint one way or another, one or another of these will stretch more or less. So that's going to be sending information to your brain about what is the exact angle of my joint. So we'll just call that like joint angle receptors. There are these Golgi tendon organs that give you like the tension on the tendons. Tendons, again, are the things that attach muscles to things. So this kind of telling you how much tension, how strong is the muscle pulling on its target. So all of these things are giving your brain information about the position of your body. If you remember when we talked about the cerebellum, the cerebellum is getting all this information. And, you know, so when you're trying to figure out, what is your body doing? What do I want my body to do? Close the loop. This is all helping your nervous system make sure your body is doing what you want it to be doing.
And proprioceptors, that's like a subcategory of mechanoreceptors?
I think of it that way, although sometimes people just use it as its own category of receptors. So either way. So now, what we're going to do now, so we're going to assume there's lots of internal receptors, and we'll look at different ones in particular as we go on through the semester. But for the rest of our discussion of the nervous system, we are going to actually just talk about the external receptors. So like the five senses. Five senses. And those are going to include, we call the general senses and the special senses. And these we've kind of introduced a little bit in the lab. General senses, again, these are like all over the body. These are the ones that include like touch, pressure, temperature, pain. They tend to have really simple receptors. Most of the receptors are, in fact, some of them, like if you're looking at temperature receptors or pain receptors, you can't even really tell what they are just by looking at them. Like if this was like the brain and spinal cord, so sometimes they're just like what we call free dendritic endings. Like under the microscope, it would just look like little branches coming off of the end of a neuron. And the only way you would know that it's a hot receptor or a cold receptor or a pain receptor is by giving it different stimuli and seeing what excites it. You can't tell just from looking at it. It's like, so they're very, very simple. Some of them are a little more complicated, like some of the touch and pressure receptors are what we call encapsulated. So they've got like the little endings, but then they've got like some connective tissue capsule around it. So maybe it's going to be better if I. So, but in general, when you think about the general senses, they're very simple. Like again, sometimes they're just little spray of dendrites at the end of a neuron. As complicated as they get are some dendrites with some connective tissue around the end. Whereas when you get to the special senses, you get really complicated things like your eyeballs and your ears and things like that. And even taste buds are way more complicated than what we see in the general senses. So the general senses, these are localized in the head. And they tend to have complex receptor organs. So, in terms of the general senses, I'm not sure I'm going to talk much more about it than I've mentioned here. Again, where does the information for like touch first start getting processed in the cerebral cortex? Anybody remember?
Prefrontal or pre-gyrus?
Yeah, it's that. So it's the so prefrontal would be more the executive, but yeah, in the frontal lobe, in that the prefrontal gyrus. So that was the place. No, actually that was motor. It was the postcentral gyrus. The postcentral gyrus was the somatosensory cortex. So postcentral gyrus. Which was actually in the parietal lobe. So the special senses, we talked about how visual was in the occipital lobe, hearing was in the temporal lobe, the other ones are in other places. So, all right, let's go to the special senses, and here we go. Yeah, maybe let's start with the chemoreception, because that's kind of the simplest one. I'm like looking at my little clock here, and yeah, I think what we'll do, I'll keep talking until like 9:30, which would be our normal thing. Then we'll take a break, and then we'll get back together after like maybe a half hour and start like lab time. But I'll continue lecturing here for about another 10 minutes. So the special senses that are chemoreception are going to be gustation and olfaction. Again, gustation, which is taste, olfaction, which is smell. Again, you've already seen these in lab, and so we know that gustation is, even though we think of it as, what we normally think of as taste is way more complicated than just taste, right? Normally, that tastes really good. As now we've seen from our lab, it's actually also a bunch about smell, it's about texture. Gustation is going to be 5 main kinds of molecules that it detects. It's going to be, for sweet, it's going to be basically looking for simple carbohydrates. For salty, it's going to be like electrolytes. Sour, it's going to be like low pH. Bitter is going to be, in general, this class of molecules called alkaloids. Again, things like caffeine is really bitter. It's a lot of the things that plants make are alkaloids. A lot of the things that plants make to protect themselves that might hurt you are alkaloids. So it's good for your body to be able to detect, oh, is there something in that I'm chewing up here that might affect me? I want to pay attention to. right? Just because it's bitter doesn't mean it's poisonous, right? Bitter caffeine is actually nice, but you want to pay attention. You know, sweet, if you're chewing something up and you're detecting simple sugars, that's like, oh, nice energy source. Same thing with electrolytes. You need, if you don't have enough salt in your diet, you're not going to have all of the ions you need to conduct all of the electrical signals for your nervous system and your muscles and all that. The receptors for gustation are taste buds. Taste buds are, we can, in fact, I'll focus. Like if you look at, you know, let me see if I can do this. Here's like somebody's mouth. Oops, sorry. Let's have their tongue. There's their tongue coming out. I'm still getting used to this. Yeah, there's their uvula. So all along the tongue are the little taste buds. The taste buds are actually on these things called papilla. So if this is the tongue, surface of the tongue, and it kind of goes down. You have these little papilla. And then the taste buds themselves are actually kind of located along the sides. Right, so they don't actually, otherwise, if they were right on the surface of the tongue, they would totally get like wiped off. So they're kind of down in these little crevasses. So the saliva can kind of come down and bathe them, and they can detect whatever's dissolved in your saliva. Right, you saw in our lab that if you put sugar on your tongue, but it didn't actually dissolve in saliva, you never tasted the sugar. The taste buds are only going to detect things that are actually dissolved in solution and kind of move over them. These little papilla are located all on your tongue, on the back of your tongue, even on your cheeks, even down your throat. You actually have some even all the way down to your epiglottis, but like 80% or something are on your tongue. So, you know, surface of it. So let's look at a taste bud in particular. So this whole thing is supposed to be the taste bud. And I've drawn a few different cells here. This is my taste cell or gustatory cell. And what do you think the mechanism is going to be for a taste bud to actually detect if there's sugar in your saliva? You've already met the main player to detect molecules like sugar. Anybody have a sense? How is a taste cell going to actually detect the presence of sugar? Anybody?
The simple sugar binds with like the receptor.
Exactly. So on these cells are going to be little receptors. Excellent. So we have basically receptors. You know, and if these were for sweet, these would be receptors for simple sugars. And when the receptor binds their target molecule, the ligand, if it's a sweet, again, it would be like, sucrose or fructose or something. Typically, it's going to open up sodium channels. And when you open up sodium channels, you'll depolarize the cell. And then there are neurons that then carry the message to the brain. So as this taste cell binds its target, it's going to open up sodium channels, which are going to depolarize the cell, which is going to ultimately trigger an action potential, which goes off to your brain. So you're saying, oh, there's sugar in my saliva. These other cells, just kind of for completeness, there's supporting cells that just kind of help anchor or stabilize the structure. And then, if you are a taste bud, it's a pretty vulnerable place to live there on the tongue, and they aren't going to live very long. Like a few weeks, things get all worn out, so you have to constantly replace these cells. So these things that I drew down at the base, these red things, these are these basal cells, and they're basically stem cells. And stem cells are just cells that can replace the other cells that are in the structure. So the stem cells, these basal cells, can divide and differentiate and become new taste cells or new supporting cells. So as the existing taste cells and supporting cells get all worn out, they get replaced by new ones. So you constantly are regenerating your taste bud. Although your sense of taste, just like every other sense that we've seen, also kind of degrades as you get older. We saw that your top frequency that you hear drops way down as you get older. We saw that your near point gets further and further away as you get older. Your sensitivity to taste goes down as you get older. Typically foods don't taste nearly as kind of vibrant as you get older and older, and people start getting more extreme with their seasonings and things like that, trying to kind of bring some pop back into their meal. So that's taste buds. Taste buds are basically detecting a certain subset of molecules dissolved in your saliva. So one of the interesting things, often if you're making bread or even if you're making candy, they have you usually add some salt to the recipe. Why would adding salt to a candy recipe, actually make it better? Does anybody have an idea? Thinking about this idea of receptors binding their target and opening sodium channels.
Would it cause a concentration gradient change?
Yeah, exactly. So if let's say the target for your receptor is sugar, and when the sugar binds the receptor, it opens sodium channels, and that creates a voltage change, which you detect as the sweetness. If you have extra salt, there's going to be a higher sodium concentration gradient, so even more sodium will come in when the sugar binds, and there'll be a more dramatic change in voltage, so there'll be a more intense experience of that sugar. So even you're not actually detecting that extra sodium as a salty flavor, it's actually intensifying the experience of the other flavor. So adding salt to things can actually intensify other flavors, because by having more sodium ions around, you get higher voltage changes due to, you know, the receptors opening sodium channels. So it's kind of fun to think about. All right, so let me turn this off here. Hold on. Stop share. So are there any questions up to this point here?
I
No.
was gonna ask about spicy foods. Like, what is it? What's going on with these spicy foods?
So that's another type of receptor. That's so it's capsaicin. I can like so this is the molecule in like hot peppers that you detect as heat, and you actually have separate receptors that bind this, and they're perceived as heat. So it's a whole other thing that's independent of what we just described as kind of taste buds. And these can actually cause heat and burning on other parts of your skin. If you've ever like touched your eyes or other sensitive parts of your body when you've got like hot pepper on, you've probably noticed that it's triggering these receptors all over. The other thing, I mean, one of the fascinating things about just kind of the evolution of like birds don't have capsaicin receptors. And what people have found is that like there's like the pepper seeds, if you eat the pepper seeds and you poop them out, your digestive tract is long enough that it actually destroys the seeds, so the seeds are no longer viable by the time you poop. But birds have shorter digestive tracts. So if a bird eats a hot pepper, by the time it poops it, the seed is still viable. So the pepper has kind of evolved this way to kind of discourage animals that have long digestive tracts from eating fruits of the pepper plant, because they would end up destroying the seeds, whereas the birds will eat the peppers, won't experience that heat and that unpleasantness, and then they'll fly, distribute the seeds around, and poop them out with some nice little fertilizer for the new little seed to start growing. So it's kind of fun to think about. So yeah, but pepper, the heat is a whole other thing. I should also mention, you know, the temperature of the food is going to affect the intensity of the flavor, because all those little channels open and close. The speed depends on temperature. You know, in general, things get slower when they're cold and faster when they're hot. So like if you're having ice cream, you actually have to make it sweeter than you normally would have if you want it to taste a certain amount of sweetness, because the coldness of the ice cream actually inhibits the response of all of those little molecules in the cell membrane that are responding to the sweet. Anyway, so any other questions? All right, so let's see. Back. Let me stop sharing. So at this point, how about how about at 10 o'clock we will regroup here. So I'm going to go offline for the next 25 minutes, and at 10 o'clock we'll regroup and we'll start talking about lab. All right. So what I am going to do right now is talk a little bit more about the special senses. I'll just finish up the chemoreceptors and then just introduce the vision, because that's what we're going to need to think about to do our next lab. So if we go into share and whoops. I'm gonna go, oh, come on. There we go. There we go. I'm gonna go into PowerPoint. I'm gonna go, okay, now I'm gonna share. And, oh, this is the wrong one. I want the other PowerPoint, which is, oh, excuse me just for a moment. I need to get into this one. Okay, the sensory. So everyone can't see this yet. So now I do new share, and we're gonna do this, and we're gonna do this. All right, so this is basically saying, you know, senses are basically, there's some stimulus, there's some, in fact, I can go into my pointer options, highlighter. Again, there's a sensory receptor, and when the stimulus hits the receptor, there's some transduction, which is going to create some electrical signal, which ultimately is going to create some kind of usually action potentials coming down the axon. This one is actually kind of interesting to look at because it helps you understand how can you encode, for instance, like the intensity of a stimulus with like action potentials. And action potentials just going like that. And yet, like, there could be soft sounds, loud sounds, but there's only an action potential as an all or nothing kind of thing. So this is actually kind of showing if you have like a weaker stimulus or a more intense stimulus, one of the ways that's encoded in the action potentials is in the rate of the firing of action potentials coming down the axon. So like if there is a quiet sound, it might be, you know, action potentials coming out at a certain rate, whereas if there's a louder sound, there might be much faster rate of action potentials coming down. So one of the ways to encode the intensity of a stimulus using action potentials down the axon is just by how fast the action potentials are firing. Like when I used to do experiments on frogs, we'd stick electrodes into their auditory nerves, and you could just hear, and then we would actually put that signal into a speaker, so you'd hear this kind of pop every time an action potential came down the auditory nerve. And when you gave them gentle stimuli, you would just hear like pop, 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 pop. When you gave them intense stimuli, sounded just like a motorboat. So this, you know, you can, you know, tonic receptors, some receptors, basically when you put the stimulus on, they kind of stay responding. You know, we talked about some things don't adapt. We talked about pain receptors don't adapt. Some of the proprioceptors don't adapt, right? Sometimes there's receptors you don't want them to adapt. You want the nervous system to know that the stimulus is staying there. But this is also really common. They're calling phasic receptor. A lot of times, you know, something responds, but then, you know, it quickly adapts and stops responding. We saw that like when you moved a hair on your skin, you had a quick response, and then pretty soon you didn't feel anything. This is just showing some of those receptors that I'm talking about in the general senses. You know, how you could just have these kind of nerve, like little dendritic endings. This might be a temperature receptor or a pain receptor. The little touch receptors are encapsulated, but still very simple. This is a root hair plexus. This is showing the, you know, detecting the bending of a hair follicle. I wonder if I can point to options, highlighter. Let's make it more intense. Blue, maybe. Yeah, so again, the simple receptors, here's like a Merkel disc, simple receptors for the general senses. This, again, this is what we saw in our lab, this idea of two-point discrimination, how if you just have one basic receptor under the skin, you can't tell the difference between 2 points or 1 point here because it's just one response. Whereas if you have a higher density of receptors under the skin, you know, then this case, like on the fingertips, for instance, you can definitely tell there are 2 points because this and this. Yeah, we don't have to do lateral inhibition. And thalamus, actually, we talked about the thalamus being kind of this relay station in the brain. Almost all your senses, with the exception of smell, which we're going to talk about in a few moments, is actually going to go through the thalamus before it goes up into the cerebrum. So we'll talk a little bit more about that in a second. And actually, this I mentioned, and I'll mention. This is an interesting thing. Looking at how pain is transmitted up into the cortex. A nociceptor here, a pain receptor, and here's this pain fiber sending some action potential down towards the brain. Actually, you know, and it goes, you know, it releases a neurotransmitter, in this case, substance P, which then talks to the next neuron and goes up into the brain, and ouch, I'm feeling pain. The opioids, things, you know, the endogenous opioids, the things that like morphine and and fentanyl and heroin and all those things would actually attach to, those receptors for the endogenous opioids are different than the ones we've been seeing in general. Normally, we've thought about the receptors being on like the target neuron, but for the opiate receptors, we actually have, let me change the color here again, highlighter, make it. We actually have the receptors here on the axon terminal that releases the neurotransmitter. So when you release the endogenous opioid, or if you're giving somebody morphine or heroin or whatever, it doesn't matter if it's endogenous or exogenous. When it binds to these receptors, it inhibits this substance P from actually being released here. So you don't release this neurotransmitter, and therefore the pain message never makes it up to the brain. So it's, you know, and like I've mentioned in class, in addition to blocking the transmission of the actual pain message up to the somatosensory cortex, the opioids also work centrally. They actually make you not care as much that you're in pain as well. So it's not just about stopping the pain message, it's also about kind of changing your attitude, you know, which kind of makes them more seductive in terms of addiction and stuff, because they can just make things not feel as bad. Again, this idea here, when you release an opioid at this terminal here, at this axon terminal, it blocks the transmission actually up. That's kind of cool. The eye, this should all look familiar. Before we do it, let's show you the la 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 hack. Oh, here. Taste bud. This is just kind of showing you let me slideshow from current slide. This is showing you the taste bud. You know, these are the gustatory cells, the taste cells with the little receptors kind of poking out into your saliva, and the supporting cells and basal cells. these sensory fibers that are sending the message to your brain when you actually do bind your ligand, like sugar or whatever. This is kind of showing what it looks like in real life. Smell is pretty much the same idea. Instead of having taste cells, you have olfactory cells. These olfactory receptor cells, though, have the same basic idea. They have receptors. Oh, this is with a mouse. This is going to look like crap. This is heck. So the receptors are on these olfactory cells that are sitting here on the roof of your nasal cavity. Rather than just binding like 5 basic tastes, olfactory cells can bind a whole bunch of different shaped molecules, and each shaped molecule has a different experience. We talked about this a little in lab. I can do this. I can show you here. Go new share. La 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 la. Oh, this is hilarious. You know, chemistry of body odors. You know, underarm odor. It's molecules that look like this and this and this. Halitosis, bad breath, are molecules actually you're detecting molecules that look like this. You know, flatulence, you're actually, you know, detecting molecules that look like this when somebody's farting. I have other, let me find some other ones here. You know, this is, let me see if I can make this bigger. You know, this is showing musk, lemon, vanilla. I've got, let me find another one here. Aroma, sense of flowers. New share, flowers. You know, if you smell a violet or a lily or a hyacinth or a lilac or a rose, it's basically your olfactory cells up in your nose are actually giving your brain information that these molecules are present in the air. So like when you sniff, you're basically pulling air with molecules up into your nasal cavity. Let me get back over here. You know, and then those molecules bind the receptors, and then those receptors send action potentials up towards your brain. You know, it's one of the things that I always think is kind of a little grody is like, again, if you like smell dog poop, it's because there are little molecules of dog poop that are floating in the air inside your nasal cavity binding here. You know, the experience of smell is your cells binding those particular molecules. So that's smell. Smell is the only of the senses that has paths up into the brain that don't have to go through the limbic system. So, you know, so when we talk about, let me just stop this sharing for a second. So one of the things that's fascinating about smell is it seems to be kind of one of the most primitive and directly wired into your brain of all the senses. I mean, again, if you think about what it actually is, it's just detecting what molecules are floating in your environment. And even if you were just like some little primordial bacteria, like in, you know, the beginning of life, you still want to know, like, oh, are there molecules that are going to be nutritious over here, or are there things noxious that I need to like start getting away from? So this just detecting what molecules are floating in the space around you, that's what smell really is. And it gets wired into your brain at, these subcortical paths up into your limbic, your emotional system and stuff in ways that the other senses are not. So any questions about chemoreception? All right, so I'm going to just start with a little bit about vision because we need to kind of bring this up in order to think about our lab for Wednesday. So let me go back to, actually not that, let's go into here. Blah, 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 blah. Okay, so vision. We've already talked about the eyeballs. In fact, oh, actually, let me go back to this thing. We talked about the eye, and then we talked about as the light comes in, it gets focused by the lens, and then the retina, which we haven't talked about in that much detail yet, which is kind of located along the back of the eyeball here. That's the place where you're going to actually do the sensory transduction, converting light into a electrical signal. The retina has two main kinds of cells in it. It's got what are called rods and cones. They get their name because of just their physical shape. The rods look more like rod-like. The cones are a little more pointy looking. The actual retina has lots of other cells that we'll talk about in more detail later. But for right now, we're going to just talk about the actual photoreceptor cells, the cells that actually convert light from, or convert light into an electrical signal. We'll back. Let's get into that later. If you look at the eyeball, when you first look straight at something, the place where the image lands here on the fovea, this is just that little spot where you look straight at something. That's going to be important in a few moments just to understand the fovea, the very center of the retina when you stare right at something. So now we're going to talk about rods versus cones in the eye. And let's do that. This whiteboard. We're going to do rods versus cones. So they're in your retina. And again, both of them are involved with converting light into an electrical signal. So they both do that. But then there are differences. One is the rods are on the periphery of the retina. The cones are in the center of the retina. That place we call the fovea centralis. In fact, one of the terms people use, like in vision research, if you're staring right at something, so the image lands on your fovea, where all the cones are, we say they're foveating. So it's even a verb. Periphery. Maybe I should, let me just rewrite that word. So so when you see things kind of more out of the side of your eye, it's more about the rods. When you're staring straight at something, it's more about cones. Again, we have cones on this side, rods on this side. Other things that are different. Cones have very high acuity, meaning that you can resolve very fine detail. Two dots that are close together still like two dots. Rods are low acuity. So you can tell something's there, but it's harder to tell exactly what it is. Normally, if we were in the actual classroom, we'd be playing around by changing the light in the room. Instead, what I'm going to do after we're done with this is I'll write up a little exercise that you can do at home and play around and actually convince yourself about some of these properties of rods and cones. But for right now, you just have to like take it as an intellectual argument. So in general, I mean, Ethan, if you put your hands to the side of your face, look straight ahead, put your hands to your side, you wiggle your fingers around, you know that you've got your hands with your fingers wiggling, but you can't really see how many fingers you're holding up if you're looking out of the side of your eye, staring straight forward. But if you put your hand right in front of your face, you can totally see how many fingers you're holding. So that's like this high acuity. You can definitely see your fingers. Low acuity, I see there's a hand, but I don't know how many fingers there are. Cones are active in bright light. So you need to have bright light for the cones to actually be functioning well. Part of the high acuity is they're looking at what's happening in a very small area of space, which means you need a more intense signal coming in to actually make them fire off. Rods are kind of integrating information over a larger distance of space, so they can actually detect lower levels of light, but they can't really tell you where it started from. So these are going to be active in dim light. Sometimes, sometimes this kind of cracks me up. People will, like, in some test question, what's the difference between rods and cones? They'll say, like, oh, cones see in the bright and rods see in the dark. It's like, nothing in the dark you can't see. What the rods do, though, is that they can actually show you images when the light level is very low. So when the light level is very dim, the cones are not going to be functioning, but the rods will still be functioning. You just won't see as much detail, but you will see there's something there. Other differences. Rods are monochrome. There is only one type of rod, and they just tell you, yes, there's light, or no, there's no light. Cones let you see color vision. The way that works is there's actually 3 different kinds of cones. You have cones that can respond maximally to red, cones that respond maximally to green, cones that respond maximally to blue. And depending on what combination of those 3 colors are getting detected, you create all the other colors. It's a little non-intuitive. If you've been used to thinking about mixing colors with paint, then, if you mix yellow and, yellow and blue, you get green if you're mixing paint and pigment. When you're mixing luminance, when you're mixing light, it's different. If in luminance, if you add all the colors in together, it actually turns white. If you add all the colors together when you're mixing paint, it turns into mud. But using these 3 different kinds of cones, you can detect all the different colors that we normally think of in the visible range. So what that means is in the bright light, you're going to see things with more acuity. You're going to be able to detect color. In the dim light, you're going to be going into rod vision, which means you still see stuff, but you're not going to see as much detail and you're not going to see color anymore. One other thing I'll mention, and this is going to be important for our lab on Wednesday, is because the rods are kind of integrating over a more amount of time, like rather than like in bright light, you can just respond. There's some bright light and I'm going to have a response. if you're trying to detect very low levels, you kind of wait and take in more information to see if something's there or not. So these respond more slowly. These have a faster response, which is going to be important in a few moments. So cones are going to respond faster. Rods are going to have a slower response. So any questions about that?
So when someone's colorblind, does that mean there's something wrong with the cones?
Yeah, exactly. So in fact, that's one of the parts of the lab we didn't do. When someone's colorblind, they have a genetic problem where they don't make one or another kind of cone. So there's different versions of color blindness. Like if you are, lacking the blue cones or lacking the red cones, you can detect some colors, but you can't tell the difference between colors that vary only by that particular color. In general, it's one of the things we call in fact, let me just start a new one. Color blindness. You say it's a sex-linked trait. Usually only in males. What does it mean to be a sex-linked trait?
It's linked to X or Y.
Yeah, exactly. So basically, this code to make the cones, this is on the X chromosome. Right, so if you have color blindness, that means you have some problem with this. So you're not making one or another kind of cone. So if you have a bad version of this X, if you're a woman, it's usually not a problem because you still have you always have two X's. As a woman, that's the definition of genetic female is 2 X chromosomes. So if you have one bad version, you still have the other version that will make the proper cone. If you're a guy and you got a bum version of the X chromosome, your other sex chromosome is a Y. So you are going to be colorblind because you cannot make that cone that you need. So it can happen. You can have a female with color blindness, but she has to have 2 bum versions of the X chromosome, whereas a guy only needs 1 bum version and he's going to be colorblind. Like in teaching this class for like 20-something years, pretty regularly we find colorblind guys in the class when we do the colorblind test, and I've never seen a colorblind woman. To test for color blindness, I can show you what that looks like. Share screen. Back to this. Pretty sure. Oh, come on, there, this. So they'll show you something like this, right? And like, if you're not colorblind, you can kind of make out, it looks kind of like there's a 29 in there. Depending on what kind of color blindness you have, you might not be able to detect the difference between the greens and the reds, and it might just look like a bunch of random dots. So it's kind of fascinating. People who have color blindness will look at the same picture as you and see something very different. We have these books in lab, and I was going to get to it, but we're not going to get to it now. But you have basically, it's pretty easy to test for color blindness by just showing people these plates. It's Ishihara color blindness test, and you can see if someone's colorblind and if they are, what kind of deficits they have based on what they see on these different special plates. So, all right, let's go back. Let's stop that. So let me see what time we got now. So for the lab on Wednesday, I'll just give you a little more. I think for today, particularly since it's our very first day online, we're gonna probably end a little early. But what I'm going to do is I'm going to just give you a little more information about our lab on Wednesday and what to expect and what you need to do to get ready for that. So let me share whiteboard. Wednesday lab, rods and cones. Obviously, part of why I've been talking about rods and cones right now is because you need to really understand the difference between rods and cones. And the other thing we need to remember from the past is the idea of a reflex. So we are going to be looking at reflexes. If you remember, reflex is just there's some stimulus. There's some integration, usually in the CNS, and there's some response. So this is your classic kind of reflex. And then we're going to think about how rods and cones might affect a visual reflex. So we're going to think about a visual reflex where the stimulus is a visual stimulus. So if we have a visual stimulus and some response we can measure, so there's going to be some time from when the stimulus happens to when we can actually see the response happen. What kind of things might affect the speed of a visual reflex from what we've been talking about?
Age.
Light,
The light.
And what about the light?
whether it's dark or bright.
Exactly. So how is that? How would dark versus right? Let's say dim versus bright. How would dim versus bright affect the speed of a visual reflex?
If it's bright, you'd have a faster response
And why exactly?
because the
Higher frequency.
Yeah.
No, higher frequency determines like the color of the light.
Ah.
Because you'd be using the cones, which are faster than the rods.
Exactly. If you're in brighter light, then the stimulus is going to be delivered into this reflex faster because cones are processed faster, respond faster than rods. If we are in dim light, then the rods are going to respond slower to feed into this, so it's going to be longer before you actually have the response happen. So that is going to be at the core of the lab that we're going to do on Wednesday. You know, the way we're going to do it is when we meet up again on Wednesday, we'll come back, we'll discuss this in more detail, and then we'll talk about how to actually design an experiment to see if that is actually supported by, you know, experimental evidence. So, you know, we'll do this all together. On Wednesday, what we'll do is we'll discuss, again, come back to this question, and then we'll look at how do you actually design an experiment, how do you think about sources of variability and try to control for them, and hopefully by then I will have figured out, in fact, I mean, I wonder if I can do this now, breakout rooms. So one of the things we're going to have to do for this on Wednesday is have the different lab groups work together. I'm going to create rooms right now. Oh, fascinating. All right, so I just created rooms, and if I start this, well, let me see. Give me a few moments here. So, because it's automatically putting you into breakout rooms, but it's not letting you choose who goes into which room. Oh, I'm going to assign manually. All right, recreate the rooms. All right, so who's like this is just an experiment for next time. So I'm going to try to put, I don't know, Rhiannon, Roxanne, and Stephen in a group in breakout room 1. Rhiannon, Roxanne, Stephen, and Victoria. They're all in a row, and I'll say add room. Oh, so are you guys in your own room? So are Rhiannon, Roxanne, or Stephen, or Victoria in the main room right now?
Yes.
Yes? Are you also in a breakout room anywhere? Oh no, so now I have to open it. So now I'm going to open it. So now, is Rhiannon still in the room? Nope. Okay, and have you guys, do the other of you see the ability to join into any breakout rooms?
No.
Okay, so that was Marissa. So Marissa, I'm going to put you in a breakout room. I'm going to assign you to breakout room 2. And Matthew, are you there? Matthew?
Yeah, I'm here.
All right, I'm going to put you in breakout room 2 as well. So now you guys are together? Okay, I'm going to close them all. All right, so is everybody back yet?
I never left.
Oh, so Marissa. Back. All right, so I think I'll figure this out. So what's going to happen is I'll put you all into breakout rooms after we have our discussion on Wednesday, and then you're going to spend some time working with your lab mates, and you will then be able to kind of actually write up. Whoa. Hello? Yeah, you'll actually write up your procedure together, and you'll be able to write it in your lab notebook, but you'll be able to talk to each other and be in your own little private Zoom breakout room. So yeah, as we go through this process, it's all going to become more and more kind of smooth and seamless. So Marissa, you're back.
Yeah, I'm back. I had to leave the room.
That's good. So what was it like being in the room?
Just hearing the other person.
Okay, so cool. So those breakout rooms are how we'll be able to have you do like little personal, or in your little lab groups, during lab time as well. So that will be a useful thing we can do. In terms of stuff to do today, you should do your best to submit your those particular spot checks for your lab notebooks using the little apps to make PDFs. I am going to make a little rods and cones exercise that you can do at home and answer and turn in sometime in, before Wednesday, but that's probably not going to show up until tonight. And otherwise, are there any other questions or comments as we kind of start this whole new world of virtual?
You said that by Wednesday we have to turn. What do we have to turn in?
You want to turn in your lab notebook, at least the scans of your lab notebook. And then you also I'm going to have a little thing that I will create at some point today that will just have you spend a little more time personally understanding rods versus cones. Because normally, if we were in lecture, I would just turn the lights off in the room so you could personally experience it. I think it's more interesting and more visceral to actually turn the lights down low and actually notice how things look different than when it's in bright light. So I'll have some little exercise that you can actually do at home. So this isn't all just kind of this intellectual exercise. It'll actually be something you can actually experience. So, but that will show up, check your Canvas module like later tonight or sometime by tomorrow morning, it will be there. So, and again, if you have any feedback or anything, please just let me know or email me. And again, I think as we continue this, it's going to get smoother and more, I don't know, more and more optimized. But right now we're just, I'm kind of glad we actually even got it off the ground here. So thank you for your patience, and I will see you all again on Wednesday morning at 8 a.m.
Thank you.
Thank you very much.
All right, stay
Thank
safe.
you.
Bye-bye.
Thank you.
Thank you.
Bye.
Thank you. Bye.
Bye-bye.
Bye.
Bye. Bye.